You're watching this with a brain. 3 pounds of soft tissue, 86 billion neurons, trillions of connections firing right now, allowing you to see, to hear, to understand these words. We've spent centuries convinced this is what thinking requires. A brain, a nervous system, complexity beyond measure. What if we were wrong? In laboratories around the world, scientists are discovering something that challenges everything we thought we knew about intelligence. Single cells. Organisms without a single neuron. Making decisions. Solving problems. Remembering. Not reflexes. Not simple chemistry. Thinking. In a petri dish in Japan, a bright yellow blob the size of a dinner plate is about to do something remarkable. It has no eyes, no ears, no brain. It is, technically, just one cell. And in the next 26 hours, it will solve a problem that took human engineers decades to optimize. It will design a railway system. This is the story of the thinking cell, of intelligence hiding in places we never thought to look, of discoveries so profound they're rewriting the textbooks on what it means to be smart. But more than that, this is a story about you. Because every cell in your body, all 37 trillion of them, may be far more intelligent than anyone ever imagined. We're about to journey into a hidden world. A world where single cells solve mazes, where organisms without nervous systems learn, remember, and change their minds. Where the very definition of intelligence is being questioned. The question isn't whether these cells are intelligent by our standards. The question is whether our standards are too narrow. In the next minutes, you'll meet a slime mold that outperforms human engineers, a trumpet-shaped creature that changes its mind, human cells that make autonomous decisions, decisions that help explain why cancer is so difficult to treat. And you'll encounter a radical new theory, that cognition didn't begin with brains. It began with the first cells on Earth. 4 billion years ago. To understand how profoundly these discoveries shake our assumptions, we need to start with what we thought we knew. For most of scientific history, the story of intelligence seemed clear. Simple organisms react, complex organisms think. The dividing line was the neuron, the specialized cell that transmits electrical signals, forms networks, and somehow gives rise to consciousness. Without neurons, there could be no learning, no memory, no decision-making. This seemed so obvious that no one thought to question it. Then came the experiments. In the year 2000, a Japanese researcher named Toshiyuki Nakagaki did something unusual. He took an organism most people would dismiss as primitive, a slime mold called Physarum polycephalum, and placed it at the entrance of a maze. At the other end, he placed food. What happened next changed biology. Physarum polycephalum is neither plant, nor animal, nor fungus. It belongs to an ancient group called the amoebozoa, and it is strange. A single cell that can grow to cover several square meters. One biological entity with thousands of nuclei, connected by a network of pulsing tubes that transport nutrients throughout its body. When it encounters food, it grows toward it. When it senses danger, it retreats. Simple behaviors, or so it seemed. Nakagaki placed the slime mold in the maze. He placed oat flakes, one of its favorite foods, at both the entrance and exit. Initially, the organism did what you might expect. It spread everywhere, exploring every corridor, every dead end, filling the entire maze with its yellow tendrils. But then something remarkable happened. Over the next several hours, it began to withdraw from the dead ends. It retreated from the useless paths. And when it was done, only one structure remained: a single tube connecting the entrance to the exit. The shortest possible path. A single cell, with no brain, no neurons, no central processing unit of any kind, had solved a maze. Not by luck, not by random chance. When Nakagaki repeated the experiment, the slime mold found the optimal solution every time. The scientific community was stunned. How was this possible? How could something without a nervous system solve a problem that would challenge a laboratory rat? But Nakagaki wasn't finished. The maze was just the beginning. He had a far more ambitious question. Could a slime mold solve a real-world engineering problem? Could it design a railway system? The answer would earn him not one, but 2 Ig Nobel Prizes, and it would force scientists to confront an uncomfortable truth. Intelligence might not be what we thought it was. Tokyo, a metropolitan area of 38 million people. One of the most complex urban environments on Earth. Its railway system is legendary, connecting 36 surrounding cities through an intricate web of lines that transport millions of passengers daily. This network took human engineers decades to design and optimize. Countless calculations, competing interests, political compromises. In 2010, Nakagaki and his colleagues asked a simple question. What would a slime mold do? They created a map. On a flat plate of agar gel, they placed oat flakes in positions corresponding to the cities around Tokyo. 36 points of food, each representing a population center. Then, at the position representing central Tokyo, they introduced Physarum polycephalum, and they watched. In the first 10 hours, the slime mold behaved like an eager explorer. It spread outward in all directions, growing toward the scattered food sources, extending its tendrils across the plate like roots seeking water. By hour 10, it had reached all 36 cities. Its yellow mass covered the entire map, a chaotic network of tubes connecting every point. But this was only the beginning. Then came the reorganization. Over the next 16 hours, something extraordinary occurred. The slime mold began to prune itself. Tubes that carried less traffic, less nutrient flow, began to shrink. Tubes that proved efficient expanded. The organism was optimizing itself in real time. 26 hours after the experiment began, the scientists photographed the result. The slime mold had created a network. And when they compared it to the actual Tokyo railway system, the one that took human engineers decades to design, the similarity was striking. The major lines matched. The hub and spoke patterns aligned. The connections followed nearly identical paths. But here's what made scientists pause. In places where the slime mold's design differed from the human railway, the slime mold's solution was often more efficient. As Nakagaki himself noted, where the two designs diverged, the differences were the result of human politics. The slime mold had no politics, no competing interests, no budget constraints. It simply found what worked. This raised a question that couldn't be ignored. How? How does a single cell with no central processing unit, solve optimization problems that stump supercomputers? The answer, when scientists finally found it, was almost more astonishing than the experiment itself. The slime mold thinks with its body. Watch a slime mold move, and you'll see its tubes pulsing. Rhythmic contractions push cytoplasm, the fluid inside the cell, back and forth through the network. This isn't random. The pulsing creates flow patterns, and those flow patterns carry information. When the organism encounters food, the tubes near that location expand. More flow moves in that direction. The tubes become highways, directing resources where they're needed most. Tubes that prove inefficient receive less flow. They shrink. They disappear. In 2021, researchers at the Max Planck Institute made a breakthrough discovery. They found that the slime mold doesn't just respond to its environment, it remembers it. Professor Karen Alim and her colleague Miana Kramer discovered something remarkable. When a slime mold encounters food, it releases a softening agent, a chemical that travels through its network of tubes. Tubes closer to the food source receive more of this agent. They become wider, softer, more efficient at carrying nutrients. But here's the key. This change persists. Even after the food is gone, the widened tubes remain. The architecture of the organism itself becomes a map of its history, a physical record of past experiences. As Professor Alim put it, given the simplicity of this living network, the ability of Physarum to form memories is intriguing. It is remarkable that the organism relies on such a simple mechanism and yet controls it in such a fine-tuned manner. The memory is stored in the tubes themselves. No neurons required. Think about what this means. A single cell can encode information about its environment into its own physical structure. It can learn from experience. It can use that learning to make better decisions in the future. This isn't the definition of intelligence we were taught, but perhaps it should be. The slime mold experiments opened a door, but they also raised a troubling question. Was this learning? Or was it just sophisticated chemistry? Was this decision-making? Or was it just physical forces finding equilibrium? To answer that question, scientists needed a different kind of experiment, one that would test whether a single cell could do something that seemed impossible: change its mind. The answer came from an unlikely place, a golf course in Manchester, England, and an organism so small you'd need a microscope to see it. In freshwater ponds around the world lives one of nature's most elegant single-celled organisms, Stentor roeselii. Shaped like a trumpet, this creature attaches itself to debris and extends its bell-shaped body into the water. Tiny hairs called cilia line its surface, beating in coordinated waves that create a vortex, drawing water and the bacteria it feeds upon toward its mouth. It is beautiful. It is ancient. And it may be capable of something extraordinary. In 1906, an American scientist named Herbert Spencer Jennings conducted an experiment. He exposed Stentor roeselii to an irritant, carmine powder, and watched what happened. What he observed seemed impossible. The creature didn't just react, it responded with a sequence of behaviors. First, it bent away from the irritant. If that didn't work, it reversed the beating of its cilia to push the particles away. If that failed, it contracted into a protective ball. And if all else failed, it detached from its anchor and swam away to find a new home. A hierarchy of responses. simple behaviors before complex ones, as if the organism was thinking through its options. Jennings published his findings. They attracted widespread interest. A single cell displaying complex decision-making? The implications were profound. And then, 50 years later, his work was discredited. In 1967, researchers tried to replicate Jennings' experiment. They failed. They published their failure, and for decades his observations were dismissed. But there was a problem. The 1967 researchers couldn't find Stentor roeselii, so they used a different species, Stentor ceruleus. It looks similar, but Stentor ceruleus prefers to swim, not attach. Of course, it behaved differently when irritated. Its natural response is to swim away immediately. They had tested the wrong organism. And for 50 years, no one noticed. Jeremy Gunawardena is a mathematician and systems biologist at Harvard Medical School. When he learned about the 1967 study that disproved Jennings, he was astonished. "I couldn't believe Jennings' work was trashed because of this shoddy piece of work someone had done," he recalled. He decided to do something about it. For 8 years, Gunawardena and his colleagues worked to properly replicate Jennings' century-old experiment. It wasn't anyone's day job. They had no grant funding. They did it because the question mattered. Finding Stentor roeselii wasn't easy. Eventually, they located a supplier in England who harvested the organisms from a pond on a golf course in Manchester. They set up video microscopy. They developed a micro-positioning system to deliver irritants precisely. The carmine powder that Jennings used didn't work well. Perhaps its composition had changed over a century. Through trial and error, they found that microscopic plastic beads triggered the response. And then, after years of painstaking work, they confirmed it. Stentor roeselii displays a complex hierarchy of avoidance behaviors, exactly as Jennings described over 100 years ago. The organism bends, it alters its cilia, it contracts, it detaches, in that order. Simple behaviors before complex ones. But the researchers discovered something else, something Jennings could never have measured. When the organism reached the point of deciding between contracting and detaching, between its last two options, something strange happened. The odds were almost exactly 50-50, as if the creature was flipping an unbiased coin. "There's no known molecular mechanism that could implement this," Gunawardena said. "Stentor roeselii has no brain, but there seems to be some mechanism that, in effect, lets it change its mind once it feels like the irritation has gone on too long. A single cell, making decisions, weighing options, changing its mind." Why would a single-celled organism need such sophisticated decision-making? Think about it from an evolutionary perspective. Before multicellular life existed, organisms like Stentor were apex predators. They navigated complex environments, they avoided dangers, they found food, they competed for resources. For 3 billion years, single-celled life was the only life on Earth. They had to be clever. Not in the way we think of cleverness, but clever enough to survive. As Gunawardena put it, organisms like Stentor roeselii have to be clever at figuring out what to avoid, where to eat, and all the other things that organisms have to do to live. I think it's clear that they can have complex ways of doing so. This wasn't just reflex. This wasn't just chemistry finding equilibrium. This was something closer to cognition than anyone had imagined possible. But the Harvard experiments revealed something even more unsettling. They showed that our assumptions about cellular behavior weren't just incomplete. They might be actively preventing us from understanding one of humanity's greatest challenges: cancer. If single cells can make decisions, what happens when human cells start making bad decisions? And what if those decisions aren't random errors, but choices? Everything you've seen so far might seem distant. Slime molds, microscopic trumpet creatures, organisms you'll never encounter in daily life. But this story isn't really about them. It's about you. Right now, as you listen to these words, 37 trillion cells in your body are making decisions. In 2022, researchers at the University of Zurich made a discovery that challenges how we think about our own cells. Professor Lucas Pelkmans and his team asked the simple question: how do individual cells decide what to do? The traditional answer was straightforward. Cells respond to signals from their environment. Growth factors tell them to divide. Other signals tell them to stop. The cell is essentially a machine following instructions. But Pelkmans discovered something different. Human cells, it turns out, don't just respond to outside signals. They integrate information from multiple sources: signals from neighboring cells, their position within a tissue, their internal state, even what phase of the cell cycle they're in. They make context-dependent decisions. The researchers called it multimodal perception, the same term we use to describe how your brain integrates sight, sound, and touch into a unified experience. Single cells do something similar. Single cells are thus able to make adequate context-dependent decisions, Pelkmans concluded, and are therefore clearly smarter than previously thought. Your cells aren't just following orders. They're assessing their situation, weighing information, making choices. This discovery has profound implications for one of medicine's greatest challenges. Think about cancer. We've spent decades fighting it with drugs designed to send one simple message: stop dividing. These treatments blast cancer cells with signals telling them to halt their growth. Sometimes it works, but often cancer cells seem to ignore the drugs entirely. They keep dividing, they spread. The treatment fails. We've called this drug resistance. We've blamed it on genetic mutations, random errors that make some cells immune to treatment. But what if something else is happening? What if cancer cells aren't just ignoring the drugs? What if they're choosing to ignore them? Remember multimodal perception, how cells integrate multiple sources of information to make decisions? In a tumor, cells exist in a complex environment. They're surrounded by other cancer cells. They're embedded in abnormal tissue. Their internal states are disrupted in countless ways. When a drug arrives with its message, stop dividing, that message is just one voice among many. And sometimes the cell's other information, its context, its state, its perception of its environment, overrides the drug's signal. The cell makes a decision, and that decision is to keep growing. This isn't failure of the drug. It's not a mutation making the cell resistant. It's the cell deciding that, given its context, the appropriate response is to ignore the instruction. Pelkmans and his team noted, such resistance to drugs is a major problem in the fight against cancer. The solution may come from taking into account the contextual cues that individual cells experience. This might sound discouraging, but actually it's a reason for hope. If drug resistance were purely genetic, purely random, we'd be fighting chaos. Every cancer would be different in unpredictable ways. But if cells are making decisions based on context, then we can potentially change that context. we can find ways to alter the signals that cancer cells are integrating. We can communicate with them differently. Understanding cellular intelligence doesn't make cancer harder to treat. It might make it easier. And this is just the beginning. Once you start looking for cellular intelligence, you find it everywhere. Immune cells that remember pathogens they've encountered before. Stem cells that sense their environment and decide what type of cell to become. Neurons themselves, which we thought were special, are really just one expression of a far more ancient capacity for cellular decision-making. But all of this raises an even deeper question. If single cells can learn, remember, and make decisions, can they share what they know? In France, a team of researchers was about to discover something that sounds like science fiction. A slime mold that can teach. Toulouse, France. The Research Center on Animal Cognition. Audrey Dussutour has spent years studying the behavior of organisms without brains. And in 2016, she made a discovery that earned international headlines. Slime molds can learn. The experiment was elegant. Dussutour and her team set up a simple challenge. Slime molds were placed in dishes with food on the opposite side. To reach the food, they had to cross a bridge made of agar gel. For some slime molds, the bridges were normal. For others, the bridges were laced with bitter substances, quinine or caffeine. Harmless chemicals, but ones that slime molds naturally avoid. The question: would the organisms learn to overcome their aversion? On day 1, the slime molds facing bitter bridges hesitated. They extended tentative pseudopods, exploratory tendrils, and recoiled. Some took 10 hours to finally cross. By day 2, they were faster. By day 6, they crossed the bitter bridges as quickly as the control group crossed clean ones. They had learned. But was this really learning? Or just fatigue? Or sensory adaptation? The researchers tested further. Slime molds habituated to quinine were still hesitant about caffeine, and vice versa. The learning was specific to the substance they'd experienced. And when the researchers withheld the bitter bridges for 2 days, the slime mold's aversion returned. They had forgotten and had to relearn. This was habituation, one of the most fundamental forms of learning, the same type of learning that occurs in sea slugs, in insects, in humans. But it had never been demonstrated in an organism without neurons. We were astonished to find out that the slime mold could learn without a brain or nervous system, Dussutour recalled. But she wasn't satisfied. She had a more ambitious question. If a slime mold can learn, can it teach? Slime molds have an unusual ability. When 2 individuals of the same species meet, they can fuse together, becoming one organism. Dussutour wondered, what happens when a trained slime mold fuses with an untrained one? Does the knowledge transfer? The experiment was carefully designed. Some slime molds were trained over 6 days to tolerate salt, another harmless but repellent substance. These were the habituated individuals. Others had never encountered salt. These were the naive individuals. Then the researchers allowed pairs to fuse, one habituated, one naive. The fused organisms were placed before salty bridges, and they crossed without hesitation. The knowledge had transferred. When the scientists watched the fusion closely, they could see what was happening. As the 2 slime molds merged, a visible vein formed at the fusion point. Within 3 hours, the naive organism had acquired the habituated organism's learning. One trained individual could educate as many as 4 naive partners. The information passed through the network without being diluted. But Dussutour pushed further still. Slime molds can enter a dormant state called sclerotia, essentially a form of hibernation triggered by unfavorable conditions. In this state, they dry up into a hardened crust, metabolically inactive. Could a slime mold remember what it learned after waking from a month of sleep? They could. Slime molds that were habituated to salt dried into sclerotia for a month, then revived, still remembered their training. They crossed salty bridges without hesitation. Memory, preserved across a dormant state that completely reorganizes the cell's structure. No neurons, no synapses, no brain, just information, somehow encoded in the fabric of a single cell. These experiments force us to confront something profound. We've always thought of learning as something that happens in brains, memory as something stored in neural connections, knowledge as something that requires nervous systems to transmit. But here, in a creature that diverged from our lineage over 1 billion years ago, we see the foundations of something that looks remarkably like learning, memory, and even teaching. This raises an inevitable question. If the building blocks of cognition existed before neurons evolved, what are neurons really? And what is a brain? To answer that, we need to meet a scientist who's proposing something radical. A theory that could unify everything we've seen and change how we think about intelligence itself.
Michael Levin is a professor at Tufts University and the Wyss Institute at Harvard. He works at the intersection of developmental biology, artificial life, and cognitive science. And he has a theory that might explain everything. Levin starts with a simple observation. Every one of us began as a single cell, a fertilized egg. From that one cell, 37 trillion cells emerged, organized into tissues, organs, a complete body. This process, called development, requires extraordinary coordination. Cells must know where to go, what to become, when to stop dividing. The embryo must solve countless problems to build a working organism. Levin asked, how do cells coordinate on such a massive scale? And more provocatively, is this coordination a form of intelligence? The answer, Levin believes, lies in something called bioelectricity. Every cell in your body maintains an electrical charge across its membrane. This resting potential isn't just a byproduct of chemistry, it's a signal. Cells communicate through changes in their electrical states. They pass information to their neighbors through specialized connections called gap junctions. In the brain, this electrical signaling between neurons gives rise to thoughts, memories, consciousness. But Levin discovered something remarkable. The same electrical communication happens between all cells, not just neurons. From the moment of fertilization, cells throughout the embryo form bioelectric networks. They share information. They coordinate behavior. They solve problems together. Neurons, Levin argues, didn't invent electrical signaling. They optimized an ancient system that all cells already possessed. Levin calls bioelectricity the cognitive glue, the medium that binds individual cells into collective intelligences. All intelligence is collective intelligence, he writes. Each of us consists of a huge number of cells working together to generate a coherent cognitive being with goals, preferences, and memories that belong to the whole and not to its parts. The evidence for this theory is striking. Consider the planarian, a simple flatworm with a rudimentary brain. Cut it in half, and both pieces will regenerate into complete worms. The half without a brain will regrow a brain. How does it know what to build? Levin's lab discovered that the instructions are encoded in bioelectric patterns. Change those patterns, and you change what the cells build. In one extraordinary experiment, Levin's team manipulated the bioelectric signals in a planarian's body. The result? A worm that grew 2 heads instead of 1. The cells weren't following a genetic blueprint. They were reading a bioelectric pattern, a kind of electrical memory of what the organism should look like. And that pattern could be rewritten. Levin's lab went further. They took skin cells from frog embryos and arranged them in new configurations. Without any genetic modification, just by placing cells in novel arrangements, the cells began to behave in novel ways. They called these creations xenobots, living robots assembled from ordinary cells that could swim, collect debris, and exhibit behaviors their parent species never displays. Even more remarkably, some xenobots learned to replicate themselves, something frog skin cells never do in nature. What does this mean? It means the intelligence of cells isn't rigidly programmed. Cells can adapt. They can solve problems they've never encountered. They can form new kinds of collective behavior when placed in new contexts. Intelligence, in Levin's view, isn't something that exists only in brains. It's a spectrum, a continuum that starts with the first cells and extends through all of life. Each of us made the remarkable journey from mere matter to mind, Levin writes. Starting life as a quiescent oocyte, just chemistry and physics, and slowly, gradually, becoming an adult human with complex metacognitive processes, hopes, and dreams. We didn't acquire intelligence when our brains developed. We began as intelligence, as problem-solving, goal-seeking cellular collectives 4 billion years ago. This vision of intelligence as a fundamental property of life might seem abstract, but it has practical implications. If cells are intelligent problem solvers, how should we treat disease? How should we think about healing? And what does it mean for the future of medicine? For most of medical history, we've treated the body like a machine. When something breaks, we fix it. When a signal goes wrong, we block it. When cells misbehave, we destroy them. But what if the body isn't a machine? What if it's more like a mind? Michael Levin proposes something radical. We should communicate with our bodies, not control them. Drugs and other interventions should be developed as communication messages, not low-level controls, he argues. The focus for therapeutic discovery must shift to understanding how proto-cognitive systems interpret interventions. In other words, cells aren't passive recipients of chemical signals. They're agents that interpret information and make decisions. Successful medicine might need to persuade cells, not overpower them. Consider wound healing. When you cut your skin, a complex cascade of cellular behaviors activates. Immune cells rush in, clotting factors form a barrier, skin cells begin to migrate and divide. This process involves countless decisions by individual cells. They assess the damage, they coordinate their responses, they know when to stop. What if we could enhance this natural intelligence? What if we could give cells better information, clearer signals about what needs to be repaired? Levin's lab has shown this is possible. By manipulating bioelectric signals, they've enhanced regeneration in species that normally don't regenerate well. They've guided cells to build structures they wouldn't normally build. The cells already know how to solve problems. The key is giving them the right information. Return to cancer. If cancer cells are making decisions based on their context, their internal state, their environment, their perception of signals, then treatment might need to change their context, not just bombard them with stop signals. This is a different approach from traditional chemotherapy. Instead of trying to kill cancer cells directly, you might try to reintegrate them into the body's information network. Some researchers are exploring exactly this approach with promising early results. Even aging might be reframed. Levin has proposed what he calls a cognitive theory of aging. As organisms age, their cells may gradually lose the coordinated goals that kept them functioning as a unified system. The bioelectric patterns that told cells, "This is what we're building, this is how we should behave," may degrade over time. Aging, in this view, isn't just accumulating damage, it's a loss of collective intelligence, a forgetting of purpose. And if that's true, the solution might involve restoring information, not just repairing molecules. These ideas stretch beyond medicine. If intelligence is a fundamental property of life, present in every cell, in every organism, then we need to rethink our relationship with the living world. The slime mold solving mazes, the stentor changing its mind, the cancer cell deciding to resist treatment. These aren't diminished forms of cognition, lesser versions of what happens in brains. They might be different expressions of the same underlying capacity: life, thinking. We've spent centuries drawing sharp lines. Intelligent here, unintelligent there. Conscious above, mechanical below. These discoveries suggest the lines were always blurred. Intelligence didn't appear suddenly when brains evolved. It was there all along, waiting to be recognized. Which brings us back to where we started. Back to you. You've been listening with a brain. That much is true. But your brain is made of cells. 86 billion neurons, each one a descendant of ancient single-celled organisms that learned to solve problems, remember experiences, and make decisions billions of years before the first brain existed. Your neurons didn't invent intelligence. They inherited it. And it's not just your neurons. Every cell in your body, the skin cells protecting you from the world, the immune cells patrolling for invaders, the stem cells waiting to repair damage, carries this ancient capacity. They sense their environment. They communicate with their neighbors. They make decisions that keep you alive. 37 trillion intelligences working together. You are a collective. In a way, you're not so different from the slime mold. Physarum polycephalum is one cell with thousands of nuclei, connected by a network of pulsing tubes, solving problems through the collective behavior of its parts. You are trillions of cells, each with one nucleus, connected by blood vessels and nerves and chemical signals, solving problems through the collective behavior of your parts. The scale is different. The principle might be the same. We don't have all the answers. We don't know exactly how a slime mold encodes memory in its tubes. We don't understand the molecular mechanisms that let Stentor flip a coin when choosing between behaviors. We can't yet explain how bioelectric patterns store information about what a body should look like. These mysteries remain. But that's not a failure. That's the frontier. Every mystery is an invitation. Every unanswered question is a door that hasn't been opened yet. Right now, somewhere in the world, a researcher is watching a slime mold solve a new kind of problem. A lab is testing whether cells can learn things we haven't imagined. A scientist is proposing a theory that will change everything again. The story isn't over. It's barely begun. What does it mean for intelligence to be fundamental? Perhaps it means the universe is stranger and more wonderful than we knew. That consciousness, in some form, might be woven into the fabric of life itself. Perhaps it means we should approach the world with more humility. That the organisms we dismissed as simple might have something to teach us about what it means to think. Perhaps it means the boundaries we drew, between smart and dumb, between aware and automatic, between mind and mechanism, were always stories we told ourselves. Stories that are being rewritten.
We started with a question. What is intelligence? We thought we knew. A brain, a nervous system, complexity measured in neurons. But the slime mold designing railways, the Stentor weighing options, the cancer cell making choices, the cells of your body coordinating right now to keep you alive, they suggest a different answer. Intelligence might not be something that appeared late in evolution, a prize awarded to the complex few. Intelligence might be life's oldest strategy, the ability to sense, to respond, to remember, to decide. The ability to solve problems in a universe that constantly presents them. Look at your hand. Billions of cells working together, sensing pressure, temperature, texture, sending signals, making decisions. You didn't learn to feel with that hand. Your cells figured it out. They've been figuring things out for 4 billion years. The thinking cell is not a curiosity, not an exception, not a strange outlier in a world of mindless matter. The thinking cell is where we came from. It's what we're made of. And in its ancient, patient intelligence, we might finally see ourselves. In every cell, a question. In every question, a universe. This film featured the research of Toshiyuki Nakagaki, Hokkaido University; Karen Alim, Technical University of Munich; Jeremy Gunawardena, Harvard Medical School; Audrey Dussutour, CNRS and CRCA, Toulouse University; Lucas Pelkmans, University of Zurich; Michael Levin, Allen Discovery Center at Tufts University and Wyss Institute. The slime mold, Physarum polycephalum, can be purchased from scientific supply companies for your own experiments. The universe is thinking. Are you? This is Cosmic Canvas, where we trace thought back to its origins. Until next time, remember that long before anything had a brain to think with, something was already thinking. Intelligence didn't evolve. It was there from the beginning.